Okay, good morning, everybody, and welcome to the invited SIAM lecture at the Joint Math Meetings. My name is Lisa Fauci. I'm president of SIAM, and this morning I have the wonderful pleasure and honor to introduce our speaker, Jim Keener. Jim got his PhD at Caltech in applied mathematics, and while he started out thinking he was going to work in bifurcation theory, he read an inspiring paper that pointed out that perhaps chaos theory had something to do with cardiac rhythm. And so, being a dutiful scientist, he picked up the textbook of medical physiology. And going through the medical physiology textbook, he realized, my goodness, there are so many dynamical systems here that are totally untouched by applied mathematics and applied mathematicians. That was the beginning of Jim's research trajectory. And it was also, what I would say, the beginning of the field of math biology. Jim is a pioneer in math bio. At the time, there were some mathematicians who were motivated by math biology, but now we see that this is a line of research. Since then, Jim has been on the faculty at the University of Utah, where he and others have created one of the world's most vibrant training programs in mathematical biology. He has been the SIAM's editor-in-chief of the SIAM Journal in Applied Mathematics, and especially some of the students here, and some of us who aren't students anymore, know very well his two incredible textbooks, the first in Principles of Applied Mathematics, a graduate text for beginners, and the great book by Jim Keener and James Sneed, the two Jims, on mathematical physiology. So today we're honored and happy to have Jim here to talk to us about that. Okay, making diffusion your friend. Thank you.
Thanks, Lisa. It's really an honor to be able to be here and to tell you a little bit about biology. So at a meeting like this, there aren't a whole lot of people who've had much exposure to biology, and so I'd like to start off by telling you that biology is very complex. This is what's called a metabolic map. This is the diagram of all the chemical reactions that go into yeast. And this diagram is probably more complicated than any man-made technological object. It's incredibly complex. There are hundreds and hundreds of enzymes and proteins and so on going on here. And today I want to focus on three tiny little parts of this diagram. And so the real question is, what's my whole goal? So as Lisa told you, I've been interested in mathematical biology. Biology is incredibly complex, and understanding this complexity is very, very challenging. In particular, we really like to use mathematics to understand the mysteries of life. I won't tell you about my recent history, but I've come to appreciate in new ways how wonderful and complicated life is, and trying to understand that even in a small part is a very interesting and fascinating and worthwhile challenge. So what we'd really like to do is we'd really like to provide predictive and mechanistic theories for how things work, to replace the verbal explanations that you get from your doctor. Take 2 of these and call me in the morning. We'd really like to understand a little bit better how things work. We'd like to understand emergent properties. Emergent properties are things that happen as a consequence of putting a number of parts together, and you can't predict what will happen just by knowing the individual parts by themselves. So we'd like to understand emergent properties. We'd like to discover and understand unifying principles. And in particular, mathematics is, I think, very good at this, because the idea being that there are ideas that apply in one area that might apply in another area, and so the idea of transferable principles or rules of life would be very useful if we could uncover them. And we'd really like to provide a view of how things work that's not available by non-mathematical means. And so, so that's really what we'd like to be able to do, but it's hard. And so let me start off by telling you a few facts. Usually you start off by telling your kids the facts of life. I'm going to tell you the facts of death. The facts of death are that entropy always increases. Diffusion is real. Molecules move down their concentration gradient. Things tend to spread out. Dissipation is real. We all know this. Mountains erode, cars rust, computers break, information is lost. My computer, which is now 4 years old, is not nearly as reliable as it was when I first bought it. It would be nice if over 4 years computers got better, but they don't. They always deteriorate. And so, so this is a fact of life, that entropy is a very real fact of life, that, or a fact of death, if you wish, that things always, always, always spread out. And in fact, it's a characteristic of living organisms that they have figured out how to fight this reality. In particular, in order to survive, living organisms must constantly fight to overcome the dissipative effects of diffusion. And one of the things that is unfortunate but real is, as soon as an organism dies, it deteriorates, it spreads out. Diffusion takes over. And so living organisms have found a way to fight this constant tendency to spread out, to spread out. But more than that, living organisms actually figured out a way to make diffusion their friend. And what I mean by that is they figured out a way to use the fact that molecules spread out to do something positive. And in particular, they've been able to do various tasks, including figuring out how to make signals, figuring out how to build structures, figuring out how to make measurements, figuring out how to make decisions in a variety of ways. And the real question is, how do they do this? So this is what I want to tell you about today. I want to tell you 4 short stories about how organisms have figured out a way to make diffusion their friend. And the question is, well, how do they do this? And the answer is the following. The answer is that diffusion, coupled with a variety of chemical reactions, and all these chemical reactions have positive feedback, enable organisms to make and to survive and to flourish. And so I want to tell you 4 short stories of how this works. But before I do that, I have to remind you about what diffusion is. Now I assume everyone in this room knows about the diffusion equation. So in particular, what does the diffusion equation tell us? The diffusion equation describes ways in which molecules move, mostly by a Brownian motion, but in fact, we know that if you have a single molecule and you find where it goes, it'll move in this random fashion called Brownian motion. You put a whole bunch of these things together and they will spread out. And the way in which they spread out is shown to you in this very simple simulation. So this is a stochastic simulation, and on top of that is that red curve is the solution of the heat equation or the diffusion equation. We all know that that's a spreading Gaussian, and that's what diffusion does. Diffusion spreads things out. Okay, so what else is true about diffusion? Well, we also know that diffusion means that things can move between one box and another box. So if you have a large number of molecules in one box and a small number of molecules in the second box, then in fact they will move from the box in which there's a large number of molecules to a small number of molecules, and they will do so in such a way that the flux between these two compartments is governed by this very simple rule that the flux is proportional to the concentration difference divided by the length of the channel through which they move. Now, we all know this from very basic understanding, but what I want to point out is that there's quantifiable information in this. In particular, if we were in this audience currently diffusing, We're not, but if we were, maybe at the end of the talk you'll be diffusing. And if we were currently diffusing and we were passing through that doorway and people in the hallway were currently diffusing, then the rate at which people move out of the doorway would tell me what the concentration of people in the hallway is. That's what this says. So by watching the flux between these two compartments, I have quantifiable information on what the concentration out there is. And so that's a very interesting thing that biological organisms can use, and I want to show you how they do that. The point is that flux is always from high to low concentrations. The second is that flux is decreased when the length through which things are diffusing is large. So in fact, if things are diffusing down a large hallway, then they will diffuse slower than if they're diffusing out through a small hallway. So in particular, I can measure the length of the hallway by measuring how fast things diffuse out of it. That's a way to get quantifiable information about length from the rate at which things are moving. The key observation is that flux contains information that can be assessed in quantifiable ways. So now let me tell you a few short stories. The first one is a short story that goes back to 1952 with Hodgkin and Huxley. Hodgkin and Huxley were able to produce a reaction diffusion system to describe how signals are produced primarily in nerves. They worked on the squid giant axon, and I really love this picture because there's a big difference between the giant squid axon and the squid giant axon. It's really important the order in which you get the adjectives, right? So they worked on the squid giant axon. Understand the difference? And not on the giant squid axon. So at any rate, what did they learn? Well, all cells are separated, have an intracellular space and extracellular space, and the cells are separated, the intracellular space and extracellular space is separated by a membrane. And that membrane actually, there are ions on the inside and ions on the outside, and so these ions are separated, and the consequence is that the membrane acts very much like a capacitor. So those of you who had a background in electrical engineering know what a capacitor is. It has the ability to store charge. But in fact, it regulates very carefully the way in which the ions, why do I always get this message? It regulates the way in which the ions can flow across the membrane. And so you can think of this membrane as an electrical circuit with a capacitor and some resistors. Now, of course, these resistors are a little more complicated. So there are these ion channels that conduct sodium and potassium and chloride ions. But nonetheless, you can think of it as this resistive capacitive network. And the important thing is that the sodium currents that go through there are regulated in a positive feedback way. Now let me tell you what that means. And so this picture shows that when a sodium ion goes through the membrane, it changes the voltage across the membrane because it's an ion, but that voltage affects the open state of the ion channel. In particular, the larger the voltage, the more open the channel is going to be. So if you think about that for a minute, you get some ions that flow in, they increase the voltage, that causes the channels to open more, which allows more ions to flow in, which causes the channels to open more, which causes the ions to flow in, which causes the ions, you get the point. It's a positive feedback loop. And that positive feedback loop can be described through some ODEs, some differential equations, where the capacitive plus the ionic current is balanced by the total inward current. That's and these are called the Hodgkin-Huxley equations. And you can study these Hodgkin-Huxley equations. The ODE dynamics are interesting because of this positive feedback. It turns out that this system is actually a bistable system. And I've drawn here the right-hand side of this ODE system. It doesn't look bistable, but the negative part, it actually is a bistable system, has two stable equilibria and one unstable equilibria. And so, in fact, this is a threshold system. So if you're close to the low equilibrium, you're going to stay there. But if you tweak it just a little bit, you're going to flip to the other side. And so that gives you the possibility of a threshold-dependent response to stimuli. And this is a very important feature of these networks. And so that's the ODE network. But Hodgkin and Huxley realized that these aren't ODEs. These are, in fact, nerves are spatially distributed objects. They have long appendices, they have long axons. And so to model how these things work in the context of a long axon, Hodgkin and Huxley argued that you could add a diffusion term. So you have your ODE dynamics, plus you have this diffusion, and now you want to know what happens. And in a breakthrough that was actually quite revolutionary, there are thousands of papers that have been written following this 1952 paper, but in Hodgkin and Huxley, by means of a hand calculator, calculated that this diffusion reaction equation had traveling wave solutions. Now, traveling wave solutions are solutions of invariant form that move with constant velocity. And this was a breakthrough discovery, that a diffusion equation with a reaction term could have traveling waves of invariant form and constant speed. Thousands, as I say, thousands of papers have been written about this in more recent times, and we now know that this underlies the behavior of neurons, it underlies the behavior of your heartbeat, it underlies the behavior of all calcium signaling. So when you move your muscle and there's calcium that's released in order to control your muscle, the same kind of phenomena is associated with these calcium waves. Dictyostelium, the organization of dictyostelium when they come together and form buds, is all governed by the same type of differential equation, partial differential equation, that they have traveling wave solutions. That was a breakthrough discovery. Let me tell you short story number 2. Short story number 2 goes back to Turing. Now Turing, in the same year, 1952, wrote this monumental paper that argued that it might be possible to form patterns by having two different chemicals that diffuse at different rates. So you have a fast diffusing chemical and a slow diffusing chemical, and these interact in activator-inhibitor ways, and he argued that you might be able to form patterns. And since that time, lots of people have argued that maybe the stripes on zebrafish, or the real stripes on zebras, or the stripes and patterns on shells could be explained by this mechanism. I would argue that that's really still quite unclear that this is the correct explanation for this. But I do want to show you an example of a situation where this kind of mechanism might be at work. And the mechanism has to do with cell polarization. Now, what's cell polarization? Cell polarization is the ability of a cell to figure out where to put its front, where to put its nose. In particular, cells have this marvelous property. I want to show you this gorgeous picture, a gorgeous movie. So this is a movie of a leukocyte, or a leukocyte is a white blood cell, and you can see a little bacterium sitting out there in front of the leukocyte. And let's watch what this leukocyte does. So this is what your white blood cells are supposed to be doing when you get a cold or a bacterial infection. It's supposed to track down the bacteria. And so I think that's so cool. There, got it. How did it do that? How did it sniff out where that bacteria was and so move its nose, if you wish, so it could follow that particular bacterium and finally capture it? Well, I want to show you a little bit about the chemistry associated with that process. Here's the process. Whoops. Here we go. Okay, so the blue here is a cell membrane, and outside there's a signal called cyclic AMP, and in the membrane there's a whole cascade, a huge cascade of chemical reactions in which the cyclic AMP stimulates something, which stimulates something else, which stimulates something else. And at the end of the process, there's a molecule which I've identified here in this ring as A. And A is something that's called a small GTPase. You don't need to know that. But what you do need to know is that A has two different states. A has an inactive state and an active state. And in the inactive state, it's sitting in the interior of the cell, and in the active state, it's in the cell membrane. And what this cascade of chemical reaction does is it activates the A and recruits it to the membrane. So in the inactive state, it's outside the membrane in the cytosol. In the active state, it's in the membrane, and it's possible to move back and forth. However, there's positive feedback. In the active state recruits inactive state to become active. So there's a little positive feedback loop there. Now, and it's all stimulated by this signal. Now, what are the consequences of that? Well, the consequences are as follows, that what you have is you have two compartments. You have the intracellular space, which is one compartment. You have the membrane, which is another compartment. And in both of these compartments, you have diffusion, but then you have a reaction that transfers these things back and forth between these two compartments. In a membrane, the molecule diffuses slowly, but in the cytosol, it diffuses more rapidly. So you have a diffusion reaction system, as I've indicated here, a diffusion reaction system of these two molecules, one which is in the membrane and one which is outside the membrane, and you have a transfer back and forth, but you have diffusion in which these two guys can move around. Now, what are the consequences of this? Well, the first thing you can do is you can realize that if you throw away diffusion and just study that very simple ODE system, it turns out you can reduce it to a first-order differential equation. That first-order differential equation is bistable. Now, bistability is interesting because it means that there's switch-like behavior possible. So as you have a small stimulus, you might be in a small state, but if you increase the stimulus, you're going to switch to the active state, and that switch between the active state is hysteretic. Now, the hysteresis is actually very important in this context because you want to respond to a stimulus, and if you respond to it, you want to keep the response on. In case the stimulus sort of changes a little bit, you want to keep it on because you spend a lot of energy turning that on. So you really like to have switch-like behavior in this. But now if you take that switch-like behavior and you include the PDE behavior, you very quickly find out there's very interesting behavior. So let me show you what happens. In particular, the PDE has a hysteretic response. And here I show you the response compared to the stimulus and the amplitude of the response compared to the stimulus. And what you see is there's a hysteretic response. That is, as the stimulus grows, there will be a switch in which the amplitude of the response grows, and so it tells you where to put the nose. However, if you turn the stimulus back down, the switch doesn't turn off at the same place, and so you basically have a thermostat that is able to detect this stimulus. Now, the feature of that is that this enables these guys to follow the stimulus. And here I have the stimulus response. I don't know if you remember the Far Side cartoon that had that. Anyway, here we see the stimulus response to this. Let me play it again to show you that this system is capable of following a stimuli. Well, No, I'm not going to show it again. It takes too much time. You saw it, right? You take a stimulus, and the guy's able to follow that thing if the stimulus is large enough. And so this is how this chemical reaction enables these organisms to move around in response to stimuli. Okay. So this is a Turing-like mechanism. There is no violation of physics here. everything is moving down its gradient, yet I'm forming a pattern. So how can you concentrate things when everything is moving down its gradient? That's a really interesting question. But indeed, that's what's happening. There is no violation of physical principles here. What is happening is the conversion from A to A star requires energy. So I have to put energy into the system in order to concentrate things, but yet everything is moving down this concentration gradient. So what happens is you take something A, you move it into A star. Now you've depleted A, so A wants to rush in. But of course, because you have A increased A star, it wants to diffuse away. But if this diffuses away more slowly than this stuff is moving in, you end up with a pattern. So there's no violation of physical principles here, and that's important. Third example, quorum sensing. So quorum sensing is the ability of organisms to count the size of the population in which they reside. So if you were bacteria, in particular Pseudomonas aeruginosa. So Pseudomonas aeruginosa is the #1 cause of infections in hospitals. It's also the #1 cause of problems with cystic fibrosis patients. And Pseudomonas typically live in biofilms. When they're in their planktonic form, meaning when they aren't in biofilm, they swim around freely, but they cannot be detected by the immune system. And so, in fact, the immune system doesn't do anything about them. But only when they are large enough have a large enough population do they turn on their toxins. So they are able, by making sure that they don't turn on their toxins, make sure that the population is large enough, and in doing so, they can fool the immune system into allowing them to survive. So in fact, this is what Pseudomonas do. And so the question is, how do Pseudomonas count the size of the population in which they live? If you were bacteria, I could ask you, how large is this room? How many people are in this room? And if you were bacteria, you'd be able to tell me without looking around. How do they do that? Let me show you a film that illustrates this. So this is in fact a growing population of Pseudomonas, and when they become, so they start to grow. So you see a bunch of single cells, and when they turn red, they've been dyed to show that they're now quorum sensing. So they've now detected that they're in a large enough population, and so they turn on. And so the question is, how do they do that? Well, let me give you a clue. So they produce a chemical. The chemical they produce is called Autoinducer. It also has a more complicated name. It's called homoserine lactone, which you don't need to remember if I give you a quiz at the end of the talk. But this Autoinducer is really important, because in the diagram, or the picture I have up here, you can see a cross section of biofilm. Now, biofilm is the stuff that if you haven't cleaned your shower in a while, or you look at dirty pipes, there's a layer of gunk in there, and that's biofilm. And that biofilm in the cross section is full of bacteria. And in particular, the picture on the left is a picture of the cross section of biofilm stained for bacteria. Now, the one in the middle is a mutant of autoinducer, meaning they do not produce this chemical, this autoinducer chemical. However, the picture on the right is what happens when they dump a bunch of autoinducer on them and see what happens. And dumping the autoinducer on there resurrects or rescues the wild type behavior, in that then they start to form biofilm in the normal way. So this molecule called autoinducer is the key to understanding quorum sensing. So what is happening there? So I told you I wasn't going to go through the whole diagram that we had at the beginning of the picture, but I was only going to pick out small little parts. And here's a small little part. So this small little part tells you how Autoinducer is made. So up on the high left and over on the right, you'll see some italicized numbers. Those are messenger RNA molecules. One's called LAS-R, and in the middle you see something called LAS-I. These are messenger RNA molecules that produce a protein. LAS-R produces the protein LAS-R, and LAS-I is an inducer that produces a protein or an enzyme called LAS-I, and LAS-I is the enzyme that produces our autoinducer A. And then in that diagram, what you see is that the LAS-R and the A come together to form a dimer. Now that dimer is actually a regulator of genetic activity. In that dimer then can go and turn on the gene associated with this messenger RNA, so that if you stare at that long enough, you realize you have a positive feedback loop. Positive feedback loop through this dimerization of A with R. That dimerization induces the transduction, the translation, and hence the translation of messenger RNA, which translates into protein, and so you have a positive feedback loop. So that's the chemistry associated with the production of this guy, autoinducer. So how do we model the behavior of this autoinducer stuff? Well, we have autoinducer that's inside the cell, and you have autoinducer that's outside the cell. Inside the cell, it's produced at some rate, which I'm going to denote here by F. Outside the cell, the autoinducer diffuses, there's our key word, diffuses and degrades. However, there's a transfer of this autoinducer across the membrane. And remember, the very first thing I said about the transfer, the flux of things across a membrane, is that the flux of stuff across a membrane is always proportional to the difference in concentrations. So in fact, the loss of autoinducer to the cell, which goes into the extracellular space, or vice versa, the extracellular space, the stuff that goes into the intracellular space, is governed by this diffusive term, and that diffusive term has this feature where it's proportional to the difference in concentrations. Now there's one other slight feature that I have to take into account, and that is the space in which the autoinducer lives inside the cell has a different volume than the extracellular space, and so you have to compensate for the volume difference through a density factor. So you write down these diffusion reaction equations. There are two equations and the two unknowns. Autoinducer, which is inside the cell, autoinducer, which is outside the cell, and now we want to study the system. So the easiest thing to do is to get rid of the diffusion term, the spatial diffusion, and just study this two-variable system. Now, some of my former students are sitting in the audience. I had dinner with them last night. I didn't warn them that I was going to give them a quiz, but they all know what to do now. Two-variable system, ODEs. What do you do? Phase plane. Thank you very much. Phase plane. So let's study the phase plane of this particular system. It's not hard to do. The first thing you do, of course, is you write down the nucleons. And this guy has two nucleons. They're easy to see. E versus A is some nonlinear cubic-looking kind of thing. And A versus E, the second nucleon, is a linear thing. And the linear thing, the slope of it depends on the density. You see a rho, 1 minus rho sitting there in the slope. So A versus E in the green is as a slope. And so now we want to know what are the steady states or the equilibria of this particular system, and what happens when you change the density. So when you have a low density, you can see very clearly that you have a very low concentration equilibria. When you increase the density, that slope moves up, and when you increase the density more, it moves up more. And now any of us can see that in the high density state, the unique steady state is a high concentration autoinducer state, and in the low density state, it's this low concentration autoinducer state, and in the in-between state, you have three steady states. Now, the three steady states indicate that if you go a little further and look at this, what we have is a hysteretic switch. And the hysteretic switch depends on the density of the cells. And that's really interesting, because it says that as the density of the cells grows, they're producing autoinducer, but all of a sudden this switch kicks in and it says, let's have high levels of autoinducer. But now, if the density of the cells should drop for some reason, I still keep that autoinducer on, so I still continue to make that signal. And so here we have a thermostat associated with the density of cells, and that's called quorum sensing. Now, this hysteresis is really important in all kinds of switches. The thermostat in this room is hysteretic. And the reason for that is as you change as the temperature changes, you don't want to be turning the air conditioner on and off, on and off, on and off. You want there to be a range in which you turn it on and then allow the temperature to cool off a little bit and then turn it off. And so you really want a range of hysteresis. Biological systems have hysteretic switches all over the place, and here's an example of where it is. Now, so what do we learn? What we learn is the rate at which something is dumped is an indicator of the size of the space into which it's being dumped. I think that's really interesting. So if you have one cell and it's dumping something, there's not a whole lot out there, and so it goes away. If you have 100 cells and they're all dumping something, then the cells know that there's other stuff out there because the rate at which we dump is related to how much stuff is out there. And so as the stuff that's out there builds up, the rate at which I dump is cut down, and consequently, I have a way of measuring what's out there. I often tell the story that when I was an undergraduate, I won't tell you what university, I lived in a very old dorm. And you could always tell when everyone returned to their dorm on Friday nights. And that's because the toilet's backed up, right? The rate at which things are being dumped is an indication of how many people are dumping. That's exactly what's happening here. Making autoinducer, dumping it into the extracellular space, and when lots of individuals are doing that, it backs up and tells the thing to turn on. That's called quorum sensing. So we see here that organisms are able to make a measurement because of the rate at which diffusion occurs. Let me tell you my short story, my final short story. This has to do with size measurement. So what have we done so far? We've seen signals, we've seen patterns, we've seen measurements of density. Now I want to tell you size measurement. There are lots of places where organisms have to know how big they are. These are pictures of yeast, the life cycle of yeast. So these are called Saccharomyces pombe, or fission yeast. They're long tubular structures, and as they grow, they divide when they get to a particular length. And they always divide at the same length. This is pretty remarkable that they can figure out how long they are. And so the question is, how do they know how long they are? How do they measure their length? Well, the answer is that, no surprise, there's a chemical reaction involved. So this is yet another tiny, tiny, tiny little piece of that diagram that I showed you at the beginning. The question is, how is the cell cycle controlled? When does a cell go into mitosis? When does it say, okay, it's time to divide? And what's involved is reactions in the nucleus that activate a chemical called CDC2. Now, CDC2 in its inactive form does nothing. But CDC2, when it is activated, turns on a whole bunch of downstream events. And so the turning on of this chemical, of CDC2, is really important. When you turn it on, you go into mitosis. But in its normal state, it should be off. Now, it's also it's highly regulated. And in particular, there are lots of regulatory molecules, but the ones that are important for this picture is something called WEE1. Now, WEE1 is an inactivator of CDC2. So you can figure out this diagram. WEE1 inactivates CDC2. But it's also the case that CDC2 inactivates WEE1. Now, those of you who are engineers know that negative is positive, right? So you have one thing that has negative influence on something, but that has negative influence on the other, and so there you have a positive feedback loop. So that inner diagram that I've drawn there is a positive feedback loop for the regulation of mitosis. Now, of course, this feedback loop is controlled by lots of other things. And in particular, it's controlled by a molecule called CDR2, and that is controlled by yet another molecule called POM1. Now, the fascinating thing about the POM1 is that it is spatially localized. POM1 shuts down CDR2, CDR2 shuts down WEE1, WEE1 shuts down CDC2. I don't know if you get all these things or not, but what we have is a control loop where POM1 keeps mitosis at bay. POM1 keeps mitosis at bay. So here's how it works. But the interesting thing is that POM1 is isolated, is localized to the ends of the cell. So the short story is POM1 keeps mitosis at bay as long as it's close to the nucleus. But as it gets further and further and further from the nucleus, it loses control of CDC2. And so there's the possibility of a switch or a regulation that's based on length. And so one writes down a diffusion reaction equation for this. So the diffusion reaction equation is really very basic. The diffusion reaction equation says we need to know what CDR2 is doing. POM1 controls CDR2. However, CDR2 reverses itself, so it can move into its inactive state. So it has an active state, inactive state. POM1 puts it in the active state. It naturally degrades into the inactive state. And so that's what this partial differential equation says. CDR2 is in the active state, but it degrades back to CDR2 phosphate through a diffusion mechanism. However, this regulation is taking place at the boundary. So those of you who are PDE experts will recognize that as a nonlinear Robin condition. So you have a diffusion reaction equation with a nonlinear Robin condition at the boundary. And now one would like to know what does this guy do? Okay, this is actually a fairly straightforward thing to think about. So you have the POM1, which is at the boundary. You have this network of reactions that's taking place at the nucleus, at the center. And so you want to know what's happening at the center. And so that gives you a system of a PDE with a system of ODEs at the two boundaries. What happens? Well, interestingly enough, there's an ultrasensitive switch which in the dependence of CDR2 at the center of the cell. What do I mean by that? CDR2 is in a low state when the cell is short, and it switches to a high state when the cell is long. Not only that, but if you look at what happens in the nucleus, there's yet another hysteretic switch. And the hysteretic switch is now length dependent. So that as the cell grows, the switch to mitosis takes place in a hysteretic fashion. So that, in fact, the cell is able to detect how long it is. So what we have is a length-dependent hysteretic transition to mitosis. Well, I went through that fairly quickly. I don't know if you understood any of the biology I just talked about. But what I tried to tell you, and I hope you understood, is that diffusion, coupled with chemical reactions for which there is positive feedback, enables cells to do things. What does it enable cells to do? It enables them to make signals that can move. It enables them to make patterns that can form. It enables them to make measurements of size, and it enables them to make measurements of length as things move around. And I think that's actually pretty fascinating, because diffusion, which we know only spreads things out, nonetheless, when coupled with these positive feedback reactions, enables cells to do things not only to fight diffusion, but to in fact do something very useful and very important. Now, the thing that I find fascinating as a mathematician is the mathematical description of these things all has a very common feature in that they're all talking about partial differential equations of diffusion reaction type. And so the study of diffusion reaction equations mathematically has told us about a huge range of biology. That's what I call transferable principle. And that's what I think the power of mathematics comes into play. And so what hope I've told you is that mathematics has actually told us something about how biology works. And consequently, I believe that the study of biology has led us to advances in mathematics, as well as the study of mathematics has led us to advances in biology. And so with that, I will thank you for your attention. And I want to tell you that it's actually a fairly old picture of some of our group, but that play a role in this research. And I have to tell you that Jack Dockery, who's on the faculty at Montana State, Cody Fitzgerald and Kirsten Utzi played a role in this particular project. But all these guys have been important to my work. And so I thank you very much for your attention.
Thank you, Jim. In the interest of people going out to lunch, if anybody has any questions, Jim will be at the podium for the next few minutes. So I encourage people to come up and ask him questions. Again, thank you so much for coming to this session. Thanks, Jim.